So today's talk is going to be kind of the basics of tumor immunity. If you've taken a course in immunology, you'll probably know 90% of what I'm presenting. So my name is Mike Rooney. I currently work at Therapeutics, like John said, and I work on neoantigen prediction there. You don't know what neoantigens are yet, but I'll explain it. And in Neer's lab at the Broad, where I was formerly, I worked on analysis of mutational signatures in cancer related to immunity. And Sisi is going to be co-presenting the main talk with me, and she's currently in the Hacohen lab working on neoantigen prediction as well as single-cell transcriptomics. All right, so the first part of the talk is going to be really focused on basic immunity, no tumor stuff yet. And we're going to talk about how the immune system differentiates self from non-self. And we're also going to introduce you to some of the basic cells that are part of the immune system and how they interact, including T cells, dendritic cells, and the all-important molecule MHC. For the second part, it's going to be now turning to the interaction between immune cells and tumors, how the immune system recognizes cancer, why those natural responses are typically incapable of eradicating cancer, and finally, what the newest therapeutic interventions are that are actually now successfully enabling immune responses that can control tumors and eradicate them. All right, so this first part is how the immune system is able to see. So there's this pathway. It's called the MHC Class I pathway, and all cells in your body do this, except for red blood cells. And the way this works is that all the proteins, the 20,000 proteins in your genome are, when they are translated and they eventually degrade, they are chopped up into pieces in the proteasome, here. And those pieces are short, let's say like about 9 to 10 amino acids long. They're then shuttled into the ER by the TAP transporter. Within the ER, they are loaded onto this molecule called MHC, and they sit in the top here. Then they migrate out to the cell surface, where they are now available for T cells to inspect. Now, it's just some language here. We call these peptides antigens. We also call them epitopes, and these words, peptide, antigen, epitope, all mean slightly different things, but for the sake of this talk, just assume that they're interchangeable and don't get confused if I use one versus the other. Also a little confusing that this molecule is called MHC, but if you're talking about human cells, it's also called HLA, which stands for human leukocyte antigen, so don't get confused if you see MHC versus HLA. I tried to be consistent, but I probably slipped up in a few cases. So as I said, there are 20,000, theoretically, there are 20,000 different proteins in the proteome that are potentially presented in the system. And if you cut all the 20,000 proteins into the unique 9-mers, you would have about 10 million unique 9-mers that could be presented by the system. Now, we don't think that there are actually 10 million that get presented. We think the real number is more like 10,000. So about 1,000, peptides end up being presented. And so basically there's a high level of selectivity in this process, and that's actually going to be the focus of the second talk. So previously I showed you this cartoon of MHC with the peptide kind of just sitting there on top. There's also, I should mention, this smaller gene that's called beta-2 microglobulin that stabilizes the structure. Now this top groove here, in reality, it's kind of almost like a piece of bread in a toaster or a hot dog in a bun. The peptide sits in this crevice, and the peptide has to have a sequence that's compatible with this crevice. And there's two positions that are particularly important, anchor position 1 and anchor position 2, which are typically positions 2 and 9 of the peptide. And these amino acids actually burrow inward into the MHC molecule, and you need to have the right amino acids there in order for that peptide to be able to bind in the first place. Okay, so we have the system. It's presenting all these different short peptides from the proteome, and the T cells are coming along and they're looking at these peptides. Now, assuming that this peptide here came from a normal host protein, there should be no response when the T cell encounters it. On the other hand, let's say that there's a virus present in the cell. So now there are viral proteins being expressed. They are also being degraded and chopped up into pieces in the proteasome. And now let's say a viral peptide ends up being presented on the surface of the cell. When a T cell comes along and looks at that, it's going to see something that's non-self, and it's going to initiate a cytotoxic response. It's going to secrete granzyme and perforin, which will kill this infected cell. And that T cell will go on to divide many times and search out for other cells presenting this same peptide. And this is the key system that's instrumental in antiviral immunity. If you don't have functional T cells, you will be very sick. So the kind of crazy thing is, like, what's the deal with these T cells being able to differentiate self from non-self? Because we think about it here, we have 10,000 possible self-peptides and a possibly nearly unlimited number of pathogens that you could, pathogen sequences that you can encounter over the course of your lifetime. So how is your immune system going to see a Zika virus peptide and know, oh, that's non-self? How is it programmed to be able to do that? So the way this works is that every T cell, let's say it's born with a novel randomized T cell receptor. So in the genome, we have a region called the TCR beta region, and it's set up with these little segments that are kind of like exons, but not really. They're repeating segments that are very similar in sequence, but different. And there are three main domains. We have the V domain, the D domain, and the J domain. And there are these two genes called RAG1 and RAG2, which are expressed during the development of a T cell. And they literally edit the genome and splice out a chunk such that a random D segment is brought to be next to a random J segment. So now you just have a deletion of some genome, and these things are brought together. And then there's a second step where that J segment is brought up against a random V segment. So now we have this highly variable random sequence that gets created. And I skipped over a part, which is when these are brought together, it's not a clean junction. There's actually all these random nucleotides that get inserted in the middle. So this highly random region, we call this CDR3 because it's the complementary determining region. And when this is transcribed to RNA, we also have this constant domain, which is kind of the main stem structure of the TCR brought together for the final transcript. So as I mentioned, we have TCR beta. There's also another gene called TCR alpha, which is laid out almost exactly the same way. And when these two pieces come together, you get the overall T cell receptor. And this unique sequence that's created here, that determines whether or not this TCR will bind this peptide MHC. And that level of strength is what determines recognition. So what's the point of having all these random TCRs floating around your body? It seems like that doesn't really create any information at all. Well, there's this special additional step that occurs. So all the T cells, after they undergo this VDJ recombination, they home to the thymus, where they undergo a series of tests. So the first thing that's determined is, can the TCR interact with any self-peptide to any degree? So let's say that this TCR actually has a frame shift in one of its chains, so it's not really functional. There's going to be no signaling or extremely minimal signaling coming inward to the T cell. And because of that, the T cell is going to die. And the logic here is this is a non-functional TCR. We don't need this T cell. On the other hand, the T cell might have a very strong reaction to a self-peptide. And we don't want this T cell because it's going to induce autoimmunity if it's going around in the body. So this also gets deleted. And there's also a really special thing that happens. So within the thymus, there are these special cells called the thymic epithelial cells, which are presenting these peptides to T cells. And they have a transcription factor called AIR, which is just randomly turning on genes throughout the proteome. And so although you wouldn't expect there to be, say, like liver genes expressed in these cells or colorectal genes, they actually do get expressed here. And it's because we don't want these T cells to go on to respond and react to liver or colorectal specific genes. And finally, the ideal scenario that we want is we want to have this very weak level of interaction with some self-peptides. So if this occurs, the logic is, well, this is a functional TCR, but it doesn't bind anything strongly in the host proteome, so it's not a risk for autoimmunity. But we think that it's possible that it could go on later in its life to strongly interact with some novel sequence. So once this occurs, the T cell matures and then it is allowed to go into circulation, and it will spend the rest of its life looking for a peptide that it can strongly bind. And probably most of them never encounter that peptide.
How long does that
and time?
take in terms of time from the birth of a T cell, like to get through this thymus?
Actually, I don't know that number, but a lot of these are produced early in your life. So the thymus is an organ that's pretty pronounced early on in your childhood, but at the time you're 20 years old, it's mostly kind of shriveled up. So these mostly get produced during your life. But that's not answering your question. How long does this training camp actually take? I don't know that number.
So I shouldn't think of this as happening all the time and high throughput. It's something that goes on more early in your life, and you sort of
mostly, but it doesn't completely stop. And I think that's actually kind of an open question, to what degree you're still producing new T cells, in your older age. It's not totally well defined. But yeah, it's primarily a childhood process where you develop all your T cells. Yep.
And these epithelial cells make all the splice forms of all?
So in theory, if you can read in your textbook, they're making everything. In reality, this process is not 100%, and there are gaps. So this is called central tolerance. There's another line of defense called peripheral tolerance, which makes up for any gaps that occur here.
So each T cell is only able to recognize one.
Well, not really. So there's nothing that says that it couldn't possibly recognize more than one. And it's in big field right now is defining how many peptides can a TCR actually react to, and people are finding that it can be in the thousands if you just throw tons and tons of random peptides at these things.
All right, let me re-qualify that.
Okay.
So each T cell can only express one T cell receptor.
Yes.
One unique.
Yes. Well, sometimes too, because you have 2 chromosomes, and again, your textbook will say that one is silenced, but that's not always completely true.
Got it. And once a T cell is sort of mature in some sense, does that change in some way, or?
Yeah, so there's many steps to a T cell's life, and today we're not going to go through all of that, but initially it's called a thymocyte when it first creates its rearranged locus. Then it goes to the thymus, then it's a mature, naive T cell. And then it will go around the body looking for something that it recognizes. It eventually does see something, and it receives the proper signaling that this is a viral, dangerous peptide. It will mature into a memory T cell and also differentiate into effector T cells. The memory T cells will live in your body for decades. The effector T cells will expand rapidly and attempt to clear the infection. Okay, so getting more to T cells, T cells are extremely diverse, and there's many different classes and subclasses. But at the very high level hierarchy, there are 2 types, which are CD8 T cells and CD4 T cells. My cartoons so far have all been CD8 T cells, and these interact with MHC Class I, and MHC Class I is presenting intracellular proteins, or peptides from intracellular proteins. So CD4 T cells, they have this other co-receptor called CD4, which will only interact with MHC Class II. MHC Class II according to the textbook, is only or is in charge of presenting extracellular peptides from extracellular proteins. So these extracellular proteins could be from some bacteria or viral particles or chunks of dead cells. And the purpose of this duality is to have coverage for both things that are in the cytoplasm and things that are topologically outside the cell or have been engulfed within cells. And these two classes of T cells work together to clear infections. And they're both necessary.
And the binding site for the antigen is the same in both cases, or they have different species of?
The binding site for the antigen. So these two molecules, MHC Class I and II, are pretty similar, but they're from different genes, and they have different rules. Like MHC Class I, the peptide sits very snugly in this groove. For MHC Class II, the peptide kind of flops outward. And the peptides that bind MHC Class II is much less well defined.
So they have a different repertoire.
Different repertoire, yeah. So then these are the two main modes, but there's also something called cross-presentation, which is when extracellular stuff gets into an endosome, like an interior vacuole, and then leaks outward into the cytosol, and then gets presented on MHC Class I. And this is a very important process in vaccinology because typically in vaccinology you have some exogenous antigen. You're trying to get it into dendritic cells, but to initiate CD8 responses. So there's a lot of research that goes into how you get the most cross-presentation to occur, and it's not a very well understood process. But this is often how CD8 T cell responses are originally started. Which I'll actually get into more on the next slide. So here, I'm just going to go through like a theoretical infection and the different T cells that are coordinating to make this happen. So say we have an infected cell to start. It has a virus in it. It's presenting a viral peptide. It's also, its friends are dying, so you have chunks of dead cells around. You have little pieces of the viral capsid floating around. This is draining into the extracellular fluid and into the lymph nodes. In the lymph node, you have dendritic cells, which is a class of immune cells, and they are taking up these antigens, and they're doing two things. First, they are presenting them, and here we're cross-presenting on MHC Class I, and also the dendritic cell is looking for additional signs that there's a problem. So let's say that there is viral RNA present. That's a really big trigger for dendritic cells, that there's a problem. So this will induce the dendritic cell to express markers like CD80. And so then the T cell sees both the peptide and this danger signal coming from the dendritic cell. And these two things together are what give the T cell license to divide and execute its effector function. And again, this is kind of a safeguard against autoimmunity. So once that T cell is activated, it will enact direct killing against the infected cells. We also have our CD4 T cells, which will be recognizing different viral peptides presented on MHC Class II. They also rely on this second signal to be fully activated. But rather than directly killing the infected cells, CD4 T cells just recruit friends. So one of the primary types of cells that they recruit are natural killer cells, will then go ahead and directly kill the tumor. So for any infection, you need it to resolve. It can't go on forever, or else you're going to have immune-mediated pathology. So in order to turn off immune responses, when it is appropriate to do so, we have cells like regulatory T cells, which secrete IL-10 and turn off the CD8 T cells. And we also have myeloid-derived suppressor cells and macrophages, which help resolve the inflammation and start resolve the lytic part of the immune response and start secreting factors like VEGF, which promote growth of new blood vessels and healing of the site. Okay, so that's basics of the immune system. I've skipped over lots of stuff in the immune system, like B cells. I've skipped most of innate immunity, but T cells are really the name of the game for the most part for tumor immunity. So that's why I focused on that. So now getting into the interaction with cancer. So this is my own editorial here. Tumor immunity has pretty uneven history within science. So this is non-quantitative. It's just my own impression, but the field kind of got its start with this guy named William Coley, who was taking bacteria that he was growing and injecting it into people's tumors. And he claimed that sometimes this resulted in an immune response. This, of course, never be allowed today. And I think over the subsequent decades, people tried to reproduce that, and it was kind of like maybe not working. And I think the field kind of withered a bit. And in the 70s, I think most people, as we understood cancer better, there wasn't really a clear reason why the immune system would respond to a tumor. But then in the 80s and 90s, people actually started discovering genes that seemed to be specifically only expressed in tumors and possible targets for immune therapy. They also started seeing things like MHC Class I loss in cancer, which really seemed to suggest that, well, these tumors are trying to evade the immune system. So many vaccine trials were launched in the 80s, 90s, and into the early 2000s. But the problem was that most of these didn't really work that well, and then there's a lot of, people were pretty discouraged. And then in the early 2000s, Bob Weinberg published his famous Hallmarks of Cancer review, and he completely skipped any kind of anti-tumor immunity as having a role in cancer. So that was a low point where most of the leaders in oncology weren't really buying into the tumor immunity idea. But then there was discovery of these things called immune checkpoints, which I'll get to in later slides. Then they worked in cancer immunotherapy, and then the field just totally exploded. And now every company that was previously an oncology company is now an immune oncology company and trying to find new checkpoints and new treatment strategies for immunotherapy. And so we're really in the golden age of immunotherapy right now. Okay, so how would the immune system recognize cancer in the first place? So here's kind of a summary of the main ways this might happen. First of all, there's the innate immune system. This doesn't have to do with peptides presented on MHC. These are just generic signals that there might be a problem present. The first is necrotic cell death. Cells are supposed to die in a regular, controlled way, apoptosis, but in tumors, you could have hypoxia and just large numbers of cells dying in kind of an uncleanly way, which is a danger signal to the immune system, interprets that as there possibly being some kind of infectious agent present. There's also often lots of DNA damage in tumors, and they're expressing genes associated with DNA repair. And this is also a signal to the immune system that there could be a virus present, because a lot of viruses try to insert themselves in the genome and cause similar DNA damage responses. But those are pretty non-specific, and it's hard to imagine how the immune system would chase down every last tumor cell with these kind of vague danger signals. So what people tend to be most focused on is the adaptive immune system, T cells, and their ability to recognize specific peptides. And I would say there's about 4 different main classes of peptides that people pay attention to within this field. So the first is tissue-specific proteins. So as I said before, that central tolerance in the thymus is not absolute. And it's possible that in a tumor, there'll just be so much signaling from the DNA damage or this necrotic cell death that tolerance will break down. And you'll end up getting a T cell that will react to some self-peptide. So in the specific case of melanoma, you can have T cell responses that get generated against melanin genes. And as a result, if that does happen, you will have clearance of the tumor, which is wonderful, but you'll also have clearance of any melanin-producing cell. So you'll have blotches of white skin all over your body. So the next main class is these very unusual genes called cancer testis antigens. And these were discovered kind of late 70s into the 80s, and they're these genes that are not normally expressed in adult normal cells, but they are expressed in germ cells, including the testes, and for whatever reason, they also are expressed oftentimes in cancer cells. And we think the reason why they're expressed in cancer is not because they are pro-tumor or help growth. It's really just because the tumor is dysregulated, there's hypomethylation, genes that should be silenced are not silent, and these things end up being turned on in a fair number of tumors. That being said, the issue with these is that their expression profiles across all normal cells in your body are sometimes hard to define. So could there be some rapidly dividing B cell precursor somewhere that expresses one of these in normal, non-pathogenic circumstances? Possible. It's hard to do profiling so deep to rule out that possibility. The third class is the one that I'm most excited about, which is neoantigens. This is when you have protein sequence alterations that arise because of point mutations. So now you have these completely novel sequences that could potentially be recognized as non-self. You can see that this is much more digital than these genes. Like you either have this mutation or you don't. And there's going to be no normal cells that have these mutations. So this is attractive for that reason. It's pretty clear cut. And the fourth category, which I think is a little bit more like the second one, in that it's a little gray, is aberrant splicing and post-translational modifications. There's a lot of strange things that happen in tumors. Phosphorylation goes awry, splicing factors can go awry, and these would all lead potentially to novel proteins being produced. But it's just difficult to validate these, again, for the same reason. Like, just because you see something weird in a tumor doesn't mean that it's not expressed in some normal circumstances. So there's just a harder road to being sure about these guys. So I'm going to now get into therapeutic approaches that people have tried. Most of these relate to targeting one of the previous types of antigens that I just described. And the first one is cancer vaccines. So this was kind of the rage, like I would say like in the 90s. A lot of people were trying this. The general concept would be, let's take some cancer testis antigen, something that we think should only be presented in tumors. And then we'll also add what's called adjuvant, which is something that looks like viral RNA or a bacterial product that will induce this dendritic cell to think there's an active infection occurring. The vaccine is meant to target these dendritic cells, which hopefully will present pieces of this protein on their surface as peptides, and also communicate to the T cells that there's a problem present. And if all goes well, these T cells will then go and kill any cell presenting these peptides from NYS01 in this case, and you'll have clearance of the tumor. Here, the tumor, you can see, it's expressing NYS01. So these generally didn't work, and I think people were really confused about why they didn't work. As I said, probably one of the reasons is that they're not so digital. Like there is expression of these genes in normal tissue sometimes. MAGE-A3, which was a target that a lot of people were interested in, I believe is also expressed in the brain, which is a problem for a couple reasons. You're going to have some tolerance you're going to need to overcome. If you do break the tolerance, you could have some pretty bad toxicity associated with that. So that's definitely been an obstacle in this field. The other is that when people were working on these, they had no idea about immune checkpoints. And I haven't described immune checkpoints yet, so you're in the same boat as people in the 90s. But it turns out there's an additional layer to this that you need to understand in order to get productive responses.
Right.
Yep.
Did they check if the tumors actually did express?
That's the other thing, too, is these genes, as they're not critical for function, and oftentimes there's a large chunk of tumors, if you really profile, they don't really express the genes. Okay, so then one of the new advances that started working really well within tumor immunotherapy just completely bypasses MHC, and it's kind of a trick. So what people did is they took a T cell receptor, and they took the internal domain, which signals inward to the T cell, chopped off the TCR part, and replaced it with what's called an antibody domain. And antibodies are these things that can be made to bind almost any molecular surface. So they designed this antibody to bind a common receptor that is expressed on B cells as well as leukemia cells being malignant B cells. So this receptor is CD19. So here, this T cell is going to bind and kill any leukemia cell and any B cell in the body expressing CD19 and kill them. And this is an effective therapy. It works extremely quickly. Within a few days, you have clearance of the cancer. It's also quite dangerous because this immune response is so potent. Oftentimes, these patients have fevers that can put their life at risk. They need to be very closely monitored with immune suppressive agents in case they're necessary to tone down that immune response. The key here is that just so happens that you can live without B cells, somewhat surprisingly. So these patients will go the rest of their life without leukemia, but also without B cells, which are actually a relatively important part of the immune system. But these people are okay. They are immunocompromised to some degree, but they can survive. But you can see that this isn't easily extendable to other cancers. Like we can't go after a colorectal receptor because you can't live without a colon. So the main obstacle here is finding appropriate targets that are extracellular. And generally speaking, there hasn't been much traction in getting this approach to work in solid tumors. But trust me, like many people are trying to figure out how to make it work. So then, finally getting to immune checkpoints. As I mentioned, there's several different ways these immune responses can be toned down. And I think the one that people kind of knew the most about was regulatory T cells. They've been understood for a while. And there's also these myeloid derived suppressor cells and macrophages, which can suppress the immune response. But the thing that ended up being really key, which was discovered not too long ago, are these receptor-receptor interactions, which occur between CD8 T cells and the cells that are presenting peptide to them. And one of the key ones is the interaction between PD-1 and PD-L1. And what's happening here is the tumor doesn't know that it's cancer. It just thinks it's a normal cell, and it doesn't think it's infected either. So when the T cells are coming around harassing them, they're saying, well, let's tone down this immune response. And this is part of the normal peripheral tolerance that your body has to prevent autoimmunity. But what ended up being really a brilliant idea was to block this interaction. So that's what immune checkpoint blockade is. People develop antibodies that prevent the signaling from occurring. And with this simple intervention, you can rescue the function of these CD8-positive T cells. They become active again and killing their targets. And I think that I don't think it was obvious to people that this was going to work so well, but it ended up really being a game changer in a lot of different tumor types. Some question?
How do you target it to avoid a dangerous autoimmune response?
Well, I'll get to that. You can't as precisely as you would like. But I'll talk about that in a few slides. So these therapies started working in indications that people really thought of as being really hopeless tumor types, like metastatic lung cancer. Compared to chemo, where virtually all the patients would die at 2 years, we had this segment of patients, about 25 to 30%, who appeared to be completely tumor-free at that 2-year time point, which is just, maybe this, I don't know if this seems like a small change to you, but this was like huge in the field to have anything that would work for these patients. And it didn't just work in lung cancer, it worked in melanoma, it was working in bladder cancer, it was working in non-Hodgkin's lymphoma, it was working in renal cancer, and so everyone kind of just pulled all their attention on this, understanding how can we make this more effective, what are the other checkpoints that we could try tinkering with, and this is where every oncology company became an immune oncology company. They all had to develop their programs for how they were going to try to get their own drugs that have similar mechanisms. As it turns out, there's not just, it's not as simple as PD-1 and PD-L1. There's all these other receptor-receptor interactions that can be inhibitory or stimulatory. And some of these have been known for a while, but not really fully understood what their mechanisms are. Some are relatively recently discovered receptor-receptor interactions. So there's a ton of interest in understanding how all of these coordinate together to generate successful anti-tumor immune responses. And there are so many trials going on against using these types of approaches. So here, the colors represent different checkpoint medicines, which are targeting either PD-1, PD-L1, CTLA-4, which is another checkpoint gene, TIM-3, LAG-3. There's all these others that you can potentially target. And around the edge here, you see all the different indications that these therapies are being tried in. And this figure is already 2 years old. I'm sure today there's more than twice as many trials going on tackling different indications. Okay, so, but there are limitations to this, and they're kind of interesting. So one thing we notice is that for most indications, it's about 25% of patients who respond. They respond fantastic. They have what seem to be complete responses. But then there's this other, like 75%, who don't really seem to have much benefit, and it's not clear like what the distinction is there. The other thing is that was pointed out, there's not a high level of specificity here. So you're turning these T cell responses back on, but there could have been a number of T cell responses that were suppressed for good reason. But then you reverse that, so you can get autoimmunity. And some of the most common manifestations of that are gastritis and rashes, which can be potentially life-threatening. And then the other thing was that, like 2 years ago, everyone was like, okay, we just need to combine these things now. Combine PD-1 blockade with CTLA-4 blockade. We're going to bump this up to 50% complete response. And people thought that was what was going to happen, but now it's looking more and more like this just increases the toxicity of the drugs without really increasing the therapeutic benefit. So people are trying to figure out how to overcome these obstacles. Now, one really interesting thing that was observed in the course of these trials finishing is that the fraction of patients who seem to have a response is very strongly associated with the number of mutations that are in the tumor. And furthermore, if you look within a certain tumor site, like lung cancer, and stratify patients by how many mutations they have, those with a very high number of non-synonymous mutations seem to have a much better prognosis on checkpoint blockade than those who have very few mutations. So based on this, people have been thinking more and more about neoantigens, because that would be a reasonable explanation for this. More mutations, there are more protein differences between the tumor and normal cells, more opportunities for T cells to respond to the tumor as non-self. So backing this up a little bit more has been work from Ton Schumacher, who is over at the Netherlands Cancer Institute. He was able to take patient melanomas and directly extract the T cells and find T cells that he could very clearly demonstrate are responding to mutated peptides in that patient's tumor. Then there's this other researcher who is another big leader in the field, Bob Schreiber, who took mice with sarcomas and was able to sequence those tumors, rank mutations by how immunogenic his team thought they would be, and develop a vaccine targeting those mutations. And these vaccines were capable of clearing tumor in those mouse models. So with this evidence, the emerging concept that people are getting increasingly excited about is the idea of doing this for patients on a personalized basis. So in this model, you would start with a cancer biopsy. You would do whole exome sequencing and RNA sequencing to define what all the mutations are. You would then define the protein coding changes that would occur, rank them according to how likely they would be to produce peptides that would bind the MHC molecule, and then actually create a custom vaccine for that patient based on those mutations and administer that over some, maybe 2-month time period. So I work at Neon Therapeutics, and this is kind of the concept that we had when we started about 2 years ago, and we weren't aware of anyone else who was doing this. But already now, 2 years later, there's probably like 10 or 12 other companies with a very similar scheme in mind. So this is a very hot field right now. is trying to see if we can actually make this type of approach work. So in the next presentation, in the main talk, we're going to be talking about how we can predict neoantigens. Obviously, we need to be really good at ranking the mutated sequences if we're going to be able to produce a successful and effective therapy, such as a vaccine. Sisi and I will co-present the work we did on getting better at those predictions. And also, Sisi will present results from a new antigen vaccine trial using the concept I just presented that was conducted over the course of the past 2 years at Dana-Farber. And that was just published in Nature this summer. So we're excited to share that with you guys as well. So that's it for the primer this morning, and I guess we'll reconvene in 15 minutes. Any more questions? We'll go back and forth a bit during this presentation. Okay, so yeah, as John mentioned, I'm Mike Rooney. I'm at Neon Therapeutics right now. I work on neoantigen prediction, formerly in the Hacohen lab, and Sisi currently in the Hacohen lab, working on neoantigen prediction as well as single-cell transcriptomics. As was discussed in the previous slide, we're really interested in neoantigens. Normally, T cells, they're looking throughout the body for non-self peptides. And the normal circumstance in which this would occur would be if there was a virus present, making the viral peptides would be recognized as non-self. But what has been recently appreciated in cancer is that when there are mutations in proteins, these novel sequences can be presented to T cells on the MHC molecule. And these novel sequences can be recognized as non-self. And it really seems like these are kind of the mechanism behind the therapeutic successes of checkpoint therapy. It seems that what T cells are recognizing in productive anti-tumor immune responses is typically actually these mutated peptide antigens. So this has led to this concept that we could have vaccines targeting neoantigens. Now, because every patient has a completely different set of mutations, very little overlap, this has to be a personalized treatment. So according to the scheme, you would need to start with a patient biopsy, do DNA and RNA sequencing on it, call the mutations, determine the novel protein sequences, then actually synthesize with machines those mutated peptide sequences and combine that with what's called adjuvant to make a vaccine. Adjuvant's a second molecular signal that says this is something bad. You should worry about this peptide that you just observed. And it's necessary for a successful vaccine. Okay, so for today's talk, we're going to talk about how we've tried to get better at predicting which mutated peptides, of all the mutated peptides that are present, are the ones that are best to target with the vaccine, because practically speaking, we can only probably target about 20 different peptides with the vaccine. And then we're going to, within that, we're going to talk about, what's been really important is the discovery of peptide processing rules. And we're also going to talk about how we've trained new neural networks to be even better at predicting these peptides than previous state-of-the-art tools. Then we're going to talk about this trial called NeoVax, which was conducted the past 2 years at Dana-Farber, which is a personalized neoantigen vaccine trial. It was a small academic trial, had about 6 patients, but it was published this summer in Nature, and we're pretty excited to share the results of that with you. So as I showed in the primer, here's the MHC Class I presentation pathway, and we can see that if there is a mutation in a protein, that could potentially find its way to be presented on the cell surface. It needs to be chopped up in the proteasome, makes these peptides that are at 9 to 10 amino acids long, shuttled through the TAP transporter, loaded onto this molecule, then it needs to stably reside here on the surface. Now, just kind of informatically, how would we think about this? Well, say we have a proline where there previously was no proline, some other amino acid. In order for this to be a good neoantigen, we need the protein to be expressed well in the first place. We need there to be cut sites in this vicinity such that the proteasome can produce this 9-mer peptide. And we need the sequence of this 9-mer peptide to have amino acids that will make it suitable for binding the MHC molecule. And the most important positions typically are position 2 and 9 of the peptide for determining that binding capability. So these are all things that we want to consider. And what I should say is that while we are most interested in mutated peptides, we're taking a broad view that we don't need to necessarily learn the rules for mutated peptides. We just need to learn the rules in general for endogenous peptides from the proteome, and those will just translate to mutated ones. There won't be anything special about the mutated ones, we don't think. Okay, so through the primer and everything so far, I've described MHC as if it was one gene, but that's not actually the case. There are 2 different types of MHC, MHC Class I, MHC Class II, which I briefly showed previously. We're focused on MHC Class I for today's talk, just to keep things simple. And MHC Class I is actually 3 distinct genes that are all very similar to each other. Adding even more complexity to this, each of these genes is highly polymorphic, meaning that there are many versions of HLA-A in the human population, many versions of HLA-B, and many versions of HLA-C. And these versions are all, well, for the most part, they're known and documented and have numeric accessions associated with them. So to give you a flavor for this, here's HLA-A, the allele frequencies for this gene. The most common one, which you will hear about often if you read immunity papers, is HLA-A0201. It has an allele frequency of about 27% in the US population. And all the other alleles, A0101, A0301, are progressively less common. And there's this pretty long tail of very rare alleles. So if you count all the ones that are rarer than 1%, that you'd have about an allele frequency of like 6%. So this is very distributed, and it's even more so for HLA-B, which the most common one only has an allele frequency of about like 14%. So, and it's even flatter as you get out here. So there's a very high level of diversity. So what's the implications of this? So for any individual patient, you will most likely have 2 different HLA-A alleles, one from your mom, one for your dad, 2 different HLA-B, and 2 HLA-C. And so if you have a hypothetical patient looking at their HLA-A and their HLA-B alleles, this hypothetical patient has A0201, A2601, B07, and B51 for its 2 HLA and 2 HLA-B alleles. And each of these has a different preference for what peptides it will bind strongly to. So here, HLA-A0201, which is the one that is very highly studied, it prefers peptides that have a leucine in position 2 and either a leucine or a valine in position 9. Here, for instance, the HLA-B07 allele, it really wants proline to be in position 2 and leucine in position 9. So the set of peptides that will be on the surface of this patient's cell are defined by these allele binding preferences, these peptide binding preferences. So naturally, whenever you were doing prediction, the first thing you need to know, what is the HLA type of the patient? So how do we know what these binding profiles are in the first place? The main thing driving this is what the peptide MHC binding assay. It's a biochemical assay where you incubate peptide with a radiolabeled beta-2 microglobulin, an MHC. And I'm not going to go into like the nitty-gritty of how this experiment works, but the main point is that it gives you a continuous scale value for the binding strength between the peptide and the MHC molecule. And somewhat counterintuitively, a small number, say like 10 nanomolar, really strong binder. A big number, like 50,000 nanomolar, is a very weak binder. So it works a little bit opposite of what you'd think. And this experiment is relatively low throughput. However, it's been around for 20 years, so there's actually a fair amount of data that exists from this. So that data is stored in a database called the Immune Epitope Database, IEDB, which we'll refer to during this presentation quite a few times. And it's also used to build predictors, and the most famous predictor is one called NetMHC. It was developed by Morten Nielsen, who is in Copenhagen, and there are many kind of other algorithms that also designed to do the same thing. I think his is the standard, NetMHC. But the main point is that these are trained on data from this particular biochemical assay. So the thing that we have been pretty excited about is a very different approach for learning peptide binding rules, which is based on mass spectrometry. So in this context, instead of starting with purified MHC, ex vivo, you're starting with living cells, which are actively presenting peptide on their MHC molecules. You lyse those cells, and then you add an antibody, which pulls the peptide MHC complexes out, and you have a purified mixture of peptide MHC. You shoot that into a mass spectrometer, and the mass spectrometer reads out the peptide sequences for you. The mass spectrometry is actually much more complicated than that, but I'm just going to leave it at that. So we are able to find the collection of peptides that are presented on these cells. Now, this was developed also quite a while ago in the 1990s. Back then, the mass spectrometers weren't quite so precise, so you would get maybe like 10 to 100 peptides in the experiment. But in the past 5 years, the mass spectrometers have become way more precise, and so now it's relatively routine for these experiments to yield 2,000, 5,000, and some. I think the most I've ever seen in a paper is like 10,000 peptides in a single experiment. But this data is different, I should point out, in that it's Boolean, right? Like you either observe the peptide or don't. The previous data was continuous scaled, so you could kind of say, well, maybe this is not as good. But there's a few reasons why we think that mass spectrometry could be actually really important for getting us better at prediction. And the first thing is that we can have this integrated prediction capability. So previously, it was just this thing that was happening, this biochemical assay. It was, does MHC bind this peptide? But we know there's a lot more going on here. We know that, well, the expression of that source protein is going to matter a whole bunch. We know that there's some efficiency that these peptides are transported into the ER. That probably contributes. There's some efficiency that these things are chopped up into pieces. There's, of course, the binding affinity, which is a piece that we probably know the most about. And then there's also the stability of these peptide MHC complexes on the surface. So all these things have to work to a certain degree for the peptides to be presented and available for T cells, but we don't really have a good sense for how these things all weigh together. We don't have a good sense for what the precise rules are for processing. So we think that, well, since these peptides have been through all these steps, we can kind of infer from the collection of peptides what these relative rules are. The next thing is unbiased discovery. What's nice about the mass spec assay is it's sampling from the entire proteome. On the other hand, in the immune epitope database, people typically only bother to do that peptide MHC binding assay. Since it's a low throughput assay, they only typically do it for peptides they think are going to bind in the first place. If they think it's hopeless, they won't bother. So as an example of this, we can look at HLA-B4403. It's the 5th most common HLA-B allele in the United States. There are many measurements for it in the immune epitope database. And if we look at the ones that are strong binders, they have a pretty well-defined motif. But if we look at all the ones that were shown to be non-binders, that weakly bind MHC, you see the motifs look basically the same. So all the peptides that people are trying for this allele look very similar. So you see there's a very high chance that you could miss an important motif since there's so much bias in what people test in the first place. And the next thing is scalable data collection. The mass spec assay, as I said, gives you thousands of peptides in one shot. And we have a lot of territory to cover because there are still pretty big gaps. For HLA-B, for instance, 40% of patients in the US population will have at least one of their 2 alleles be one that's very poorly covered in the immune epitope database, fewer than 10 peptide examples of binders. In HLA-C, it's even worse. 50% of patients will have an allele where people really don't know very well the binding preferences. So we need something that we can quickly march through all these rare alleles and define these binding preferences so we can treat all patients. So the team that worked on this, the PIs were Nir Hacohen, Steve Carr, and Kathy Wu at Dana-Farber. Sisi and I worked closely together. Jen did the mass spec work. She's now with me at Therapeutics. And Chrissy and Darren, they worked on kind of taking care of the cells and the biochemistry. And Carl Klauser who was on the 5th floor of the Broad, worked on the mass spec aspects. the data search aspects of the mass spec. So coming to this assay, there was one little wrinkle that we saw, which is that even though we can potentially learn all these things from the mass spec data, we have this problem, which is that the cell isn't presenting just one allele, it's presenting two HLA-A alleles, two HLA-B alleles, and two HLA-C alleles. So when we do the experiment, we're gonna have this hot mess of peptides that bind different alleles. We're not gonna really know for sure which peptide maps to which allele, which is kind of a pain in the data analysis. You know, our goal in this is to really cleanly and finally map the binding preferences of these alleles, and that's not gonna be so easy if we have 6 different alleles present simultaneously. So the trick that we used is that we started with a cell line called B721.221, which just so happens not to have MHC expressed because of a deletion in its genome. We could then add in a single allele of our preference via a plasmid, and then get a cell line that's expressing a single allele. And in doing this, we have this nice clean population of peptides that all bind a known allele. So we did this for 16 different alleles, HLA-A alleles and HLA-B alleles. Some of them are very common ones where we knew very well already what the binding preferences was, so we could have that as validation. We also included some rare alleles so we could define those binding preferences kind of de novo. So among our peptides, we just want to do like a reality check. Do they match what the MHC, the pre-existing predictor, says? So do our hits look like binders? Do random peptides look like non-binders? And for most alleles, this is what we saw. We saw very clearly that real mass spec peptides score strongly. Remember, a low number is a strong binder. And that decoy peptides had poor predicted binding. But then there are other alleles where it wasn't so clear-cut. So here's one of the rarer alleles. It would seem, based on this, that the pre-existing motif was incomplete, and that we were completing that motif. But of course, we want to test the idea. Maybe there's something wrong with the mass spec. We want to see, are we discovering bona fide new motifs in the data? So Sisi is going to speak to that for a bit.
Thank you. Excellent. So this is around the time when I actually joined the team. And with this large, new, exciting mass spectrometry data set at hand, we started asking some questions, like we wanted to know how good is the quality of our data set? How does it agree with the current pre-existing peptides that are already deposited in the immune epitope database that Maya described? And do we, for example, recapitulate existing biology that's already known for well-characterized binding, well-characterized motifs for alleles like A0201? And more interestingly, we wanted to know, does our data add new value? So we started doing that with looking at basic sequence logo plots for the motifs that bind every allele. So here I'm showing a logo plot for all peptides that were characterized in the mass spectrometry dataset for A0201, and that's compared to the motif based on peptides that were found in the immune epitope database. And as you can see, for the anchor positions at 2 and 9, the motifs agree really well. So just very quickly, in case you're not familiar at these logo plots, the idea is that the y-axis, or the height of each position, represents the the information content, and then within each bar for each position, the letter is also scaled according to its frequency. So here we see that leucines are found both at position 2 and 9 in each one of the 2 data sets. Okay, so that's encouraging. But also interestingly, we tend to see that there are more pronounced signal for inner positions in the peptide, like for example here in position 6 and 7. And these could potentially be sub-anchors that were not appreciated before. And I'm showing A0201 here, but this was really the case across each one of the 16 alleles that we profiled. So here is like a bird's eye view of comparing the frequency of amino acid at the amino acids. So these are in columns for every allele in the rows, and then across every position of 9 more peptides. So these are these horizontal blocks for position 1, position 2, and so on in position 9. The cells that are colored in scales of blue color correspond to residues that would be enriched in the IDP database as compared to mass spec. And then for the orange colored cells, well, those will be enriched for in our mass spectrometry data set. Some particular examples include methionine and cysteine, which were overrepresented in IDP, and part of this is actually something that has to do with how we sequence the peptides out of the mass spectrometer, and we can account for later on. And then isoleucine and valine, for example, are overrepresented in the mass spec data. Oh, so here's. So this is great, but we've only looked so far at frequencies of residues at every position, and we actually want to do better than that. So, for example, what if we know that certain residue is present as an anchor at position 2? Does it tell us anything about the preferred anchors at position 9, for example, or are they totally separate from each other? So to do that, we wanted to develop a method to cluster and visualize groups of similar peptides. The first thing we need to do this is to develop a function, essentially, to score the distance between two peptides. So given peptide 1 and peptide 2, we want a number that says how similar or different they are. One way to do this is by using amino acid residue similarity matrix, like, for example, BLOSSOM. But we also wanted to take into account the specific knowledge of our domain problem space. So, for example, we know that anchor positions are more important for binding. So if two peptides agree or disagree in an anchor position, this difference is more important than a difference in some kind of one of the internal positions that are mostly noisy. So to do that, we decided to give a higher weight to anchor positions, and then we also wanted to make sure that we don't bias what anchor positions are. So this point is important because even though most alleles do have anchors at positions 2 and 9, there are actually cases where the most pronounced anchors are elsewhere on the peptide. Like, for example, A0101 has an anchor at position 3. So in order to account for that, we just define weights based on entropy. And finally, we define our metric just simply as the average of the entropy-weighted distances between the residues of each position when you scan through the two peptide sequences. Right, so we have two peptides, and we scan across from position 1 through position 9. We get the similarity between every position based on the similarity matrix. And then we scale that number by the entropy at that position.
What similarity matrix are you using?
So, good question. So, we are not using BLOSSOM. We are actually using a similarity matrix that was HLA binding specific. So, it was derived from data that was HLA binding specific. And it's called the PMBEC matrix, and you'll see that later on as well.
Does this satisfy the triangle inequality?
I don't know. But it works as a distance function. Okay. So we can call it a distance function if that makes you happy. So if you take an allele and you line up all of the peptides, so it'll be the entropy of residues at each one of the positions of the peptide.
Sometimes the HLAs bind like 10-mers and 11-mers and stuff. Like, how do you do this when the epitope is a different length than 9?
We didn't show this, but the mass spectrometry data allows us to actually do a de novo preference, the distribution of preference of different lengths for peptides that bind. And we do see that most peptides are indeed of length 9. So we developed this for peptides of length 9. But you can extend it, as long as you have enough observations for other positions, you can apply the same. Yep. So for the analysis, I'm mostly going to show peptides of length 9.
So that 1 minus entropy term will end up concentrating.
So
the largest contribution to the sum will be on what end up being the anchors. But
right, exactly. And again, we wanted to do that because if two peptides have the same anchor, they're most they're more similar in this particular problem. Like they're more likely to bind the same allele, whereas if they're the same in some kind of an internal noisy position, it doesn't really give us much signal. Okay, so then we went ahead and we computed a big pairwise distance matrix for all the peptides that were found in our data set. And then we actually append to that the peptides that are found in IDB. So this is just a small snippet of the matrix. In order to be able to eventually visualize that, we had to reduce the dimension, and we used non-metric dimensional scaling here. And then finally, we can use our 2D representation to plot. So in these plots that I will show, every circle is a peptide, and the color represents the source of the peptide. So the peptides that came from our mass spec data are colored in orange, and the peptides from the IDB data set are colored in blue.
So you used multidimensional scaling to turn the pairwise distances into an embedding in low-dimensional space.
Yes. And now we can ask very particular questions about what are the binding motifs of each particular cluster of peptides. So if you look at that well-pronounced cluster in the middle here, we can look at the logo plots, and for the MS data and for the IDB data, we can see that we have leucines at both anchor positions, which makes sense. This is one of the well-known binding motifs. But a more interesting example is this different allele 2902, where you have a smaller cluster, which is fairly well represented in the mass spec data. We have 19 peptides with that motif, but that had only one single peptide from the IDB with that same motif. So this is, again, to the point that mass spectrometry is an unbiased way of collecting peptides. And now we have more data to be able to interpolate that as a binding motif, whereas from IDB, you wouldn't be able to do that because you just don't have enough support for that cluster. And another way we used the visualization technique was to explore our binders that we selected for experimental validation. So what we did here is we wanted to ask, how do we know whether the peptides that we think are very strong binders are actually binding motifs that are real? So we went ahead and we selected peptides to actually synthesize and validate for binding with the binding assay, in vitro binding assay that Mike described. And the way we selected peptides is, first we scored all peptides with a mass spec-based models, and we took the top 10 percentile, so the most highly confident hits. And then we looked for any of those peptides that were actually in the bottom 10 percentile that when scored by the NetMHC algorithm. So I'm showing here the peptides for one particular allele, B5101, highlighted in orange. So with this strategy, 11 peptides were selected for validation. And in terms of the predicted binding affinities, these are all very, very large numbers. Remember, large numbers means a poor affinity, whereas in fact, they all validated, this is the experimental measurement now, to be a very strong binders, all except one. And when you visualize them on this 2D map, we can see that some of these peptides land in clusters that really don't have support in IDB. So it's normal that the algorithm, the neural network won't be able to learn those binders. So now back over to Mike for the next part of the analysis. Yeah. Oh.
Sorry, go ahead.
Yeah.
So the clustering that you have, do IEDB and your mass spec prediction of binding affinity tend to agree within a cluster?
Yes.
Like does the similarity that you plotted there seem to explain binding affinity as well? Like if two things are close by in those space, would we expect them to either both bind well or not bind well?
So I mean,
not necessarily.
within a cluster, the motif is essentially
same.
the same, but what you'll tend to see is that there'll be some clusters that are very mass spec heavy. Most of the examples are mass spec peptides, and like one or two are from IEDB. And so within those, if it's the case that it's dominated by mass spec and couple IEDB, what you would typically see if you run that NetMHC prediction, and it's a very poor prediction. It's not a sequence that NetMHC, trained on IEDB, expects to be a good binder. But what we show here is that if you take those and measure them, they are in fact very strong binders.
So within one cluster, they're mostly going to be binders, not necessarily all with higher affinity, but yeah.
Maybe I misunderstood. Don't you have, shouldn't some clusters be poor binders? Don't you have all potential peptides?
We only,
Hillary,
this
do
is
you mind
only a
repeating?
plot that shows only binders.
Sorry, it's just hard to hear. Do you mind repeating that?
Sorry, I was just clarifying the plot is only of binders. It's not all peptides with this new distance matrix. It's only peptides that should be binding.
Well,
and
so
then looking
mass spec generates only binders because
Yeah.
that's what you observe in the binders. And IED
Right.
contains things that validate as binders as well as things that aren't binders. And these plots will only show the things that were validated as binders.
Okay, thanks.
So you-
Sorry, thank you.
The yeses from IDB and the mass specs, which we know are yeses.
Yes. Okay.
And there are also weird things like INDB, like I think you were showing that as well. There's some peptides that appear in multiple experiments, and sometimes they'll be binders, sometimes they won't be. So you kind of have to filter those out.
So I'd like to, can we skip the
Yeah.
questions till discussion? Because I just want to make sure we get through the rest. So the next thing that we were really interested in is the processing question. So each peptide is coming from a source protein. So there's some source sequence context around it. And we were curious whether that sequence context was contributing to whether or not the peptide was likely to be observed. So there was a previous effort to do this back in 2005. It's the same group that created NetMHC. They created an algorithm called NetChop that was trained on about 500 data points. And they derived this motif, which we thought looked a little funny. Like it didn't really correspond to enzymes that we would expect to be present. And generally within the field, people regard this algorithm not to really be predictive. And even the person who wrote it doesn't suggest using it. So we thought, well, we have all this new mass spec data now. Maybe we could write something or derive something much cleaner and more precise. So what we did is we looked at our mass spec peptides and we looked at the frequencies of amino acids upstream and downstream of them. So in this plot, U1 means one position upstream of the peptide, U2 is 2 positions upstream, etc. D1 is 1 position downstream, etc. And the frequencies were compared to the frequencies that we would observe upstream and downstream of random decoy peptides. And these random decoy peptides were selected from the proteome, but matched according to the first 2 and last 2 amino acids of hit peptides, just so we recreate the same sequence biases that might exist. And learn a motif that's truly specific to processing and not just like, non-random sequences that exist in the proteome. So some of the strongest things that we observe are, first of all, this strong enrichment of basic residues, arginine and lysine, just upstream of the cut site. And this corresponds to an enzyme that we know is active in the proteasome, which is trypsin, which cuts just after arginines and lysines. But then what was pretty, I think, unexpected was this enrichment of arginine and lysine downstream of the cut site. And we don't actually know what process produces that. We don't know if it's an initial tryptic cut followed by chew back on the C-terminus of the peptide, or some other enzyme that hasn't been clearly identified yet. The other pattern that we see is this depletion of proline. So the blue means, we see proline in these positions much less frequently than we expect. And this corresponds pretty well to what we know about proline. It creates a kink in the peptide chain, and a lot of different enzymes that are proteases have a difficulty cutting at or near prolines. But then another thing that was less expected was this enrichment of alanine. There are some enzymes associated with cutting near alanines, but maybe not so much associated with the proteasome specifically. But this is another very strong signal that we see in our data. So we saw a collection of both expected and novel enrichments in these data that were pretty strong, extremely significant, but also like magnitude-wise quite strong as well. So then we looked at the frequencies of these amino acids upstream of the peptide, within the peptide, and downstream of the peptide. And we saw that cleavable amino acids like alanine, arginine, and lysine, their frequency is higher just upstream and just downstream of the peptide, but within the peptide, they're depleted. And proline, which we saw seemed to be interfering with cleavage, it's depleted around the cut site, but within the peptide, proline seems to be enriched. So what we think this means is that successful peptides that end up being presented, not only have they been cleaved in the right site, they've avoided cleavage internally. So we have depletion of cleavable residues and enrichment of poorly cleavable residues within the peptide. And then we were curious, well, we see this processing signature in the particular cell line that we happen to be using for these experiments, but is this a general signature? Would we see this in other cell types as well? So we looked in previously published mass spec data from breast cells, colorectal cells, fibroblasts, HeLa, donor blood cells. And across the board, we see most of all the same signatures present across these distinct cell types. So it seems that this is actually a fairly conserved proteasomal processing signature and not highly specific to our cell line. Though we do see some points of variation which we're pretty curious about. For instance, the relative enrichment of arginine and lysine upstream, which was quite strong for some cell lines, but then nearly absent for some cell types such as fibroblasts. And we think this probably has to do with variable expression of certain proteasomal enzymes. One more reality check was to look at Class II peptides. It's also possible to use these mass spec protocols for MHC Class II. We expect these to have a very different processing signature because it's a completely different pathway. And in fact, that's what we see, a very different signature. So it wasn't just some kind of artifact of the way we were doing the analysis. It's completely different. And then we finally compared the signature against what we would have expected based on the most highly ranked and low ranked peptides according to the net CHOP algorithm, the previously existing standard. And while we see some relationship here, there's a lot of things that are different. So what we've produced is a pretty big update on the processing rules of the proteasome. So the next thing we were curious about is gene expression. And within the field of MHC peptidomics, it's still a pretty common idea that the MHC peptidome is decoupled from the RNA transcriptome. More recently, people have chipped away at this and shown there actually is a pretty decent statistical relationship between RNA transcript expression and whether peptides are presented. But still, it hasn't been clear how you would use this when you're doing neoantigen prediction, for instance. Like, do you just use some cutoff, or is there some weighting of the two that you could apply to have better prediction? So what we did is we drew a grid, and that grid was according to expression level and predicted affinity according to that net MHC pan predictor. And we binned all our observed mass spec peptides and counted how many fit in each bin. We also took a very large number of random 9mers from the proteome, and we binned those as well. So the top number in each grid cell is the number of mass spec hits. The bottom number is the number of random proteome 9mers that fell in that bin. Then, having those two numbers in each bin, we calculated the ratio, and that's what the red to blue coloring here is in the plot. And what we see is that there's actually a fairly decent multiplicative relationship between these two variables. You can compensate for an order of magnitude decrease in binding strength with an order of magnitude increase in the expression level. And this is important because it means that there are these populations of peptides that are actually good candidates for a neoantigen vaccine that we would have just skipped over if you had used a threshold. And the time we wrote this paper, using thresholds was certainly what everyone was doing within the field, or just completely ignoring expression. The next thing that we are interested in is the localization of the source peptide, the localization of the source protein of the peptides. We thought, well, maybe peptides in the cytosol will present better than peptides that come from proteins in the cytosol might present better than peptides from proteins in the nucleus or secreted proteins, for instance. So we did this analysis, and we did see some pretty big differences, which we were interested in, but some of it didn't make complete sense. Like, we weren't sure why secreted proteins would be an enriched category. Like, it seems like they would actually be depleted, if anything. And what we realized is there's a big goof in this analysis is that you need to control for expression level when you do this. If you don't control for expression level, you're going to get rubbish answers. And if you do that proper correction, you see the MHC peptidome is super democratic. It's pulling from all compartments of the cell, seemingly relatively equally. So this was another important finding, because within the literature, there's a lot of confusion about whether there were localization biases, because no one was doing the expression correction, and each paper would have a different localization that was the more important one. But I think this shows that it's actually a surprisingly equal process. So then we thought, well, great, we know the expression is important. We have this nice updated cleavability predictor. We combine everything along with the affinity and have better prediction of which epitopes are most likely to be presented. So we did something very simple here. It's just a logistic regression where we're going to weight each of these and come up with a likelihood that peptides are presented. And to score this, we used maybe a little bit of an unusual approach. We, instead of using something like AUC, we used positive predictive value. And we set this up such that when we score, we have some number of mass spec hits that we score, and then 999-fold that of decoy peptides. So say we have 1,000 mass spec hits in our data set, we would then spike in 999,000 random 9-mer decoys and ask our model to rank everything, and then we would see how many of the mass spec peptides fell in the top 0.1% of the rankings. So the reason, I'll just do this slide because I'm going to go fast. The reason we do this is that AUC, so this is such a biased problem. It's kind of like a needle on a haystack. You're just trying to find these very rare binding peptides. If you use something like AUC, it's not properly tuned to that. So you can have actually relatively poor predictors. This one in red, for instance, would score just as well as something that was very sensitive to rare peptides. And so using this kind of metric creates a lot of confusion. Models that actually perform very differently in the vaccine setting would have equivalent scores. So this is why it was important for us to use this positive predictive value setup. So then we were interested in how these variables play together. So we literally just did every combination of variables, and we saw how well the resulting model did in explaining the data, looking at the positive predictive value. And you can see that A here, that corresponds to an affinity-only model. R is an RNA expression-only model. P is a protein expression-only model. C is a cleavability-only model. Naturally, you can't do anything very well at all without having affinity in your model. And then down here are all the combinations, like building gradually more complex models. And what we did is then we just asked, well, let's just add one variable at a time, add the variable that gives us the best increase in our predictive capability, and see what happens. So what we saw is that an affinity-only model is getting you about 28% on the positive predictive value. Adding an RNA expression, you jump considerably by 18 percentage points on the positive predictive value. Adding in cleavability, you add another 8% in your positive predictive value. And once you have those variables, there's actually relatively little you can do from these other variables like localization or protein expression levels. So it turns out that just having RNA-seq is really enough to define the MHC peptidome. You don't need to do protein expression level experiments. Added sequentially. So yeah, so for instance, so if you added protein first, protein expression level, it would explain a sizable chunk, not as much as the RNA expression. Yeah, but practically speaking, you're going to do RNA-seq, not proteomics, to define these expression levels. And so this is showing us that not only is that better, you don't even get much cumulative gain by adding in the two sources of data.
What were you using to determine affinity?
So this is just using NetMHC. But that brings me to the next part is and I should just say here, you can use NetChop, that pre-existing processing predictor here, and then we only give you a 3% bump, whereas we're getting an 8%. So again, we improve the prediction of peptide processing. So now we're going to get to new neural networks, not using NetMHC anymore, and defining predictors de novo of binding potential.
Okay, so just a recap of what our problem statement is. So we want to predict, given a peptide and a specific HLA allele, is it likely that the peptide is going to be endogenously presented on that allele? Some things to note is that this is a complex 3D interaction. We've seen this picture of the peptide binding groove and how the two anchor positions kind of duck in. And we also want to be able to consider the fact that there are possible synergies between different residues in the peptide sequence. And we also have seen how not only binding affinity, but also endogenous processing signals like expression and cleavability are important for observing the peptide as presented on the cell surface. So the solution we turned to was neural networks. And here is what the network architecture looks like. It's actually a quite simple model. We have 50 hidden units in one single hidden layer, and the model is fully connected. The main, novelty in this particular modeling is the fact that we do introduce these two signals, expression and cleavability, that capture endogenous processes of the peptide. And outside of that, our other sets of features include various ways of encoding the peptide sequence, as well as capturing the properties of each amino acid residues of the sequence, and then some features that capture properties of the peptide as a whole. So a note here to say that we settled on this architecture for a few different reasons. One of them being that the current net MHC models use a similar architecture. And also at the beginning of this work, our goal was to actually prove the quality of the data rather than to try to beat the model itself. So we wouldn't try to create a very complex model architecture just to say that our model is better. We wanted to say that our data really brings new value. And there's definitely a lot more work to be done here. So what are the input features? We think of them as in two main categories. Peptide intrinsic features are features that you can compute just given a peptide sequence. So for example, here we have input features that capture the amino acid sequence, and we introduced 3 different sequence encodings. I'm going to show examples of these in a minute. And then we also capture properties of the whole peptide. So for example, how long is the backbone of the peptide? What is the overall hydrophobicity of the peptide? Overall, there are 8 features like that. And then for the peptide extrinsic features, as you've seen already, we do incorporate expression in terms of the TPM value of the transcript where a certain peptide came from, and the cleavability score, which is the output of the model that Mike just described. So these are our peptide intrinsic features that capture the sequence. It's very similar binary or one hot or dummy or however you want to call it encoding. The example here shows that we have the 20 amino acids. So every time you have a match between the residue that you're featurizing, so for example, the first residue is A, it matches with A, so you have 1, 1, and 19 zeros. For the second residue in the peptide, you have leucine, which will have leucine marked as 1, and everything will be 0 and so forth. The next part of encoding was based on amino acid similarity. So rather than having a binary 0 and 1, now you have a more discriminatory score of how similar the two residues are that we are comparing. And this is based, again, on the blossom matrix that I've mentioned before. And the final encoding was analogous to this, but with a different amino acid residue similarity matrix, which was the one that's derived based on HLA binding data, which actually gives a continuous number here. So it's more of a fuzzy type of encoding.
So what are you comparing in this encoding? Is it all pairs of positions? Or?
All positions. So if you have one peptide of length 9, every residue becomes a vector of length 20,
Yes.
and you just string them all together. So I'm not comparing 2 peptides, I'm only featurizing one peptide.
You want amino acid?
I'm sorry?
You mean one amino acid? Or, sorry.
I'm featurizing one peptide by turning each residue, so each amino acid, into a vector of length 20 and stringing these vectors of length 20 all together. So you have 9 of them, so 9 times 20, 180 input features.
Right, so each amino acid becomes one hot encoding of like 20.
Yeah. Yes.
That was the first
Right.
encoding. And
Yeah.
in this one, you have for a given amino acid, you're now encoding it a second time with this string of 20 numbers, or is it something between pairs of amino acids? That's what I'm curious about.
So it's a string of numbers. And so before, when we compare, does K match L? And you say no, so it would be a 0, right? In one hot encoding.
Yeah.
Here, instead of just having a binary 0, 1, we would go to our similarity matrix and we'll say, what is the similarity score between K and L? And this will be this number.
You do that for every.
Okay. For all 20, yeah.
And the third one was derived from the distance matrix that was earlier in the top.
The third one is derived from a similarity matrix, so like a 20 by 20 residue similarity matrix. That's HLA binding data specific.
Okay.
Yeah. So then we also introduced some features to capture amino acid properties. Here, since we can think of like probably hundreds of different types of amino acid properties. What we used is analysis done by another group that tabulated all of these different properties and then we run a simple PCA analysis on that. So we used the first 3 principal components of that analysis to featureize our data. So again, for every residue, now it's encoded as 3 numbers representing these first 3 principal components of amino acid residue properties, I guess. So the models were trained with very standard 5-fold cross-validation. They performed well on internal evaluation, but it's most exciting to see how the external performance looks like. And to do that, again, a quick note to say that IDB was not a good candidate to evaluate our models because a lot of the data in IDB is derived from in vitro binding assays, while we want our models to reflect endogenous processes. So for the evaluation, we selected 2 data sets, which are also mass spectrometry based. One of them is single allele and the other one isn't. Here, each group of bars represent 1 cell line and allele combination. And there are 4 different models that are being compared side by side. The light blue and the dark blue are 2 different versions of NetMHC, the current state of the art that is predominantly being used. And the yellow and orange are 2 different flavors of the models that we've trained. The difference being that in the orange model, we did include expression and cleavability, while in the yellow one, we didn't. And across all of the different evaluations, we see that especially when you add expression and cleavability, we outperform the NetMHC models quite drastically, actually. So in the last 5 minutes, I quickly wanted to end on how does all of this analysis and thinking actually translate to something that's tangible in the clinic. So enter the NeoVax trial. This is a trial spearheaded by Kathy Wu near Hacohen and Ned Fidge. It's under the supervision of Dr. Patrick Ott, and it's conducted in Dana-Farber. So this far, 6 patients were melanoma patients were enrolled, and essentially we went through this strategy of developing a personalized tumor vaccine as the pipeline that Mike talked about earlier. Essentially, we collect tumor and normal samples from the patients, then they undergo DNA and RNA sequencing. We also do HLA typing so that we know what the patient's HLA alleles are. And then we identify the top ranked peptides that are candidates for inclusion in the tumor vaccine. The peptides are synthesized and the vaccine is administered to the patient. And this just shows that the administration includes a prime section and then 2 later boosts after a few weeks. So what's really exciting is that—
I just want to say one thing real quick. These patients, they receive surgical removal of their tumor, and at that point they're tumor-free, but it's known based on the history of these patients, they typically have recurrences within a year or 2. The objective here was to see whether this approach would prevent those recurrences from happening.
So it was really exciting to see that all patients actually do develop T cells that are specifically recognizing the introduced peptides in the vaccination. So these results show both responses against CD4 and CD8 T cells, where we have T cells that recognize the immunizing peptides, but we also show that they do not react against the wild-type version of those peptides. So essentially, these are assays measuring interferon gamma production of T cells that have been stimulating with a peptide, either the mutant version or the wild-type version, and red lines correspond to the mutant, while wild types are in black, and you see that there is no response. Predominantly responses only to mutant peptides. And then, oh yeah.
Naive question maybe, is there a danger of like reversion back to wild type? Like what's the mutation rate?
No, that won't happen.
No way.
No,
Okay.
it's probabilistically.
Yeah. Okay.
You'll lose mutations sometimes because a deletion of a part of the chromosome. So that can happen.
Yeah.
And then finally, look at how the patients are doing. So 4 out of the 6 patients are still alive and well after being tracked for 36 months when, as the paper came out, and they're tumor-free. 2 of the patients, after having administered the neoantigen vaccination, relapsed, so the tumor reoccurred. However, they were administered anti-PD-1 or checkpoint blockade that Mike described earlier, which again resulted in progression-free survival without tumor.
So, this trial, like they would have done this with checkpoint from the very beginning, like given it at the same time as vaccine, if they could have. But it just wasn't a possibility when they were putting this together. And this concept of putting checkpoint blockade together with a neoantigen vaccine is exactly what everyone wants to do now. It's what NEON is doing now. And the thought is with these 2 patients who went on to have these complete responses when the checkpoint was brought on, the thought is that that's like a synergistic effect. Typically, as I said before, only about 25% of patients who receive checkpoint have a complete response. Here we see 2 out of 2 with a complete response. Obviously, super small numbers. We couldn't do a big study, but this is to us is very encouraging that there could indeed be a synergistic effect, and larger trials are happening now to vet this.
So as the trial was initiated a while back, the algorithms that were used to select peptide for vaccination were actually the NetMHC, the existing algorithms. So in terms of next steps in a trial that's conducted in Therapeutics now, they are actually using insights from the MS analysis to select epitopes for vaccination. And I believe for the new patients in the new LAX trial, we'll be doing the same thing. And so you've seen at the primer how to initiate an immune response, you really need the first step to occur, which would be to have a peptide presented with MHC, but then later you also need the T cell to recognize that peptide MHC complex. So there are a lot of exciting research ongoing that's in the same way as we're trying to predict binding rules for peptide binding MHC, we want to also be able to predict rules for T cells recognizing peptide MHC complexes so that we can also score not just for binding, but in a sense for immunogenicity. And we'll probably be spending a lot of our efforts doing that next. Mike, do you have anything else?